part of a very exciting series, and today we're going to hear more about the animal side of things, as Wing said, and the One Health aspects of it. And it occurs to me that as the most exciting part of most of our weeks now, or even fortnights, is the trip to the grocery store, thinking about exactly what's going on in terms of the meats and the livestock becomes something that perhaps comes more into view. So the speaker today is well prepared to answer that and other questions. And he's, as Wing said, the director of the National Animal Disease Center of the USDA's Agricultural Research Service, and has 38 years of experience in infectious, metabolic, and emerging infectious diseases research. They have a very big, almost $35 million a year congressionally appropriated animal health and food safety research program. So Dr. Kehrli is going to talk to us today about coronaviruses in livestock, vaccines, and international transport of virus. So thank you very much for attending. And Dr. Kehrli, go ahead.
Okay, thank you, Phil and Wing. It's a pleasure to be with everyone today, albeit via webinar. What I want to acknowledge here is, first, I'm not a virologist, nor do I play one on TV. My background is as an immunologist. So I have obtained the assistance in part of this presentation from several colleagues, including Dr. Kelly Lager, Dr. Kay Fauberg, who are two virologists at our center, as well as Dr. Megner and Dr. Scott D, respectively from Kansas State University, and Scott, formerly from University of Minnesota, but now with the Pipestone Applied Research Veterinary Services Group in Pipestone, Minnesota. So, as many of you on this call probably are aware of, coronaviruses have been around for a very long time. And in fact, the first coronavirus ever described was back in the 1930s. That was avian infectious bronchitis virus. And it was kind of confirmed to be a virus retrospectively when they went back and examined tissues from some earlier outbreaks because the technologies for identifying viruses were evolving at about that time. But veterinarians can see a wide range of viruses. This is I'm going to call it a short list. These give you 10 examples. They're by no means in order of importance or prevalence, other than many of these viruses have been around a very long time, as you'll see. And they range in viruses that cause mild respiratory to very severe enteric infections. And the viruses on this list are restricted to their host species in general and are not considered to be zoonotic pathogens. So the presentation I'm going to give you today is I'm going to focus on a couple of examples that may not be obvious to all coronavirus people, or to people that are not familiar with coronaviruses, but there's been some evolution in the viruses that we see in veterinary medicine that have changed the tissue predilection for a virus to infect, and I'll briefly touch on that. And then talk about a virus, actually a couple of viruses that got introduced into the United States in 2013, and share with you some of the information that has evolved since that event, as to some of the things that are important in controlling viral infections in livestock. And that'll be the latter part of the talk and is the second half of my title. So what do we know about the coronaviruses? I've highlighted here in yellow on this table the viruses affecting livestock or pets and what we know about their receptors. And as you can see, all of these animal coronaviruses that are listed here actually do not use the same receptor that the SARS-CoV or SARS-CoV-2 use. So they are distinct viruses and represent no zoonotic risk, and they do not obviously then spread to people, and they are not a food safety concern. These viruses are as I mentioned, they can be lethal. Typically, the enteric viruses can affect the younger animals most severely. And I'll focus on these pig viruses, a couple of them here. The Transmissible Gastroenteritis virus. It's been around since the mid-1940s, and it's a severe enteritis It causes villus blunting, where the villi and the gastrointestinal tract are attacked by the virus and they shed, and you get a very severe diarrhea with high mortality rates, typically in the first week or two of life. Similarly, the porcine epidemic diarrhea virus, which was first isolated in the mid-1970s, that one is indistinguishable clinically from the TGE virus, but they are distinct viruses. And as you see on the slide, it suddenly was introduced into the United States in 2013. One of the things that's unique about the TGE virus is not unique amongst viruses, but it obviously has a relatively high mutation rate. And in 1984, in Belgium, a mutation was detected in the TGE virus that actually changed the tropism of the tissues that virus infected, and it became porcine respiratory coronavirus. And almost simultaneously, similar events happened and were detected in the United States and elsewhere around the globe. So TGE and porcine respiratory coronavirus are highly similar. And the big difference is that TGE replicates in and destroys the absorptive epithelial cells of the small intestine, causing diarrhea, and is fatal there. Whereas the porcine respiratory coronavirus that acquired those spontaneous deletion mutations, primarily in the spike protein, it causes a subclinical or mild disease in pigs and is mainly a mild respiratory disease. There are a few reports of a more severe, but for the most part, it is believed that it is quite mild in its clinical presentation. What was rather interesting was when this virus emerged, the belief was Mother Nature had created a mutation that almost presented it as a vaccine to protect against TGE virus. And TGE kind of became less of a problem in modern production environments. It didn't entirely go away. It still is out there, but the respiratory coronavirus did elicit some neutralizing antibodies that would reduce the severity of TGE. So there's some interesting aspects of the relationship between TGE and the respiratory coronavirus. Now, if we go on to the porcine epidemic diarrhea virus, that's one of our more recent transboundary animal diseases that has been introduced into the United States swine population. And the electron micrograph here on the right-hand side of the slide was actually obtained from a diarrhea sample from a pig submitted to Iowa State University's veterinary diagnostic laboratory. And at that time, the diagnostic tests were not picking up a virus. And so they actually brought some materials from that particular case out to our facility. And our electron microscopist was able to process the diarrhea sample and obtain this lovely picture of a coronavirus, a pretty typical set of spike proteins surrounding the core virion. And that was the first indication that this emerging disease that was not being identified as TGE or anything else was indeed a disease being caused by a coronavirus. And then very quickly, the veterinary diagnostic laboratories developed PCR diagnostic tests and confirmed indeed that this was porcine epidemic diarrhea virus. And it had extremely high nucleotide similarity to PED viruses found in China. This was first detected in April. You can see on, well, maybe you can't see on the micrograph, but the electron micrograph was taken on May 10th. So a fairly rapid ability to confirm that this indeed was a coronavirus. Now, what was impactful about this introduction was that U.S. pork producers lost about 10% of their herd within 1 year as a result of this virus getting into their herds, where 7 to 8 million baby pigs died in a very short period of time. And this virus spread rapidly across many of the major, well, all of the major pork production states in the U.S. So this was a pretty devastating impact on the pork industry, and veterinarians were struggling trying to figure out any epidemiological links between the various locations that this was appearing on farms across the United States. There were no apparent obvious movements of animals that explained everything, nor movement of people. So it was an investigative process that went on for several months. And this slide here simply illustrates that of the millions of head of hogs in 2013, this little valley here that you can see represents the pigs that were lost. And then, of course, producers were able to get more pigs bred, and they began replacing their supply. But it had a dramatic impact in that period of time. What was also interesting is that after you see the data here is for about a 5-year period ending in 2018. But you'll see here, these are new PED cases reported by week. And you can see a very clear seasonal pattern, which again is totally consistent with the cold weather in the various locations where these samples were submitted from farms, and a very obvious seasonal pattern of the cases of porcine epidemic diarrhea virus, PEDV. Curiously, something else happened at that same time, and this is a paper that illustrates how the previous occurrence of TGE virus, which was obviously very cyclical in nature, again corresponding in these peaks to the cold weather months here in the United States, a very typical pattern of when TGE was hitting our hog production systems. And it never goes down to complete zero, but typically you would see this. When the PED virus hit, it's almost like TGE went away. Now, there's no reason to believe that this is a result of any cross protection between these two viruses, but more likely what happened was the blue dotted vertical line here, at the point where producers and veterinarians figured out that we had an introduction of a transboundary virus in the United States, the pork producers and veterinarians really stepped up the biosecurity on farms and were much more careful for entrance and exit procedures, and did their level best to try to prevent accidental introduction of viruses into production systems. So there's a lot of possible contributors to how this virus for TGE began to become a very minimal problem in the U.S. Swine herd, if you will, but quite coincidental with the introduction of the PEDV virus. So PEDV virus, there were some vaccines that came onto the market, and they do help in reducing the situation in farms that have become infected and are endemically infected now with it. But the existing vaccines that are available do not currently do an adequate job of protecting a naive herd from the virus being introduced to them. So scientists, and I'm going to give an example of some research that we've had going on at our center, which involves Dr. Kay Fauberg in a collaboration with a colleague of hers, Dr. Susan Baker at Loyola University of Chicago. And they've been working on developing modified live or attenuated coronavirus vaccines. And they've developed a reverse engineering process where they've targeted a couple of gene segments in the virus that are involved with defeating the innate immune system of the host. And by that, I mean trying to interfere with the ability of the virus to block type 1 interferon production. So they have 3 different non-structural proteins that are involved in antagonizing type 1 interferon that they've gone in and targeted some mutations. They pieced the virus back together, resurrected, and then have been testing it first in vitro in isolated porcine alveolar macrophages. And then after very encouraging results demonstrating that the mutated virus fails to suppress the type 1 host interferon response, then went on and we examined it in some pigs and found some very encouraging results. So our research is currently focusing on the porcine epidemic diarrhea virus, but also working on orthologous vaccine constructs for two other coronaviruses in pigs, porcine Delta coronavirus, which was also introduced into the United States around 2013. And then a third coronavirus, which has more recently been described in China, referred to as the swine acute diarrhea syndrome virus. These all have some very similar designs in how the virus attempts to suppress the host interferon response. And so it's our belief that if we can develop vaccines that attenuate the virus, we may actually be able to get more effective vaccines, and that these may actually be delivered through the oral or oral nasal routes. So as I mentioned, they've targeted some of the non-structural proteins. They actually have a construct that has a total of 4 different distinct mutations that they've made, a total of 4 in the 3 non-structural proteins. And as I mentioned, the attenuated virus did elicit a very robust interferon response, both in vitro and in vivo. So that's all very encouraging, and some of that work has already been published. And this just outlines the study that was done. And in this study, they took 3 groups of pigs that were 4 to 7 days old, and they proceeded to expose them to the wild type virus or their mutant vaccine construct or mock infected, and then went on to monitor the clinical score, viral shedding, and then also looked at virus specific IgG titers. And on panel B here in the lower left, this is on the y-axis is a clinical score, and this rising line for the wild type virus, it was the typical situation of exposing pigs to porcine epidemic diarrhea virus, where they got a fairly severe clinical diarrheal disease within 2 or 3 days, whereas the mutant construct, those piglets did not seem to be affected in any clinical way by the mutant virus. And thus it was very encouraging that this is very well attenuated from a clinical disease expression point of view, and that the virus that was mutated, that contained those 4 mutant constructs, was no different than the mock infected pigs. And as you can see in panel C here, that the virus did replicate in these square boxes here in the lower, but it was obviously substantially reduced from the level of viral replication that we saw with the wild type virus. So very encouraging signs here, and we have more studies going on to evaluate this as a vaccine, looking at its ability to immunize pregnant sows, which will be a very important aspect of any vaccine against PEDB, because it affects pigs at such a young age that you need to get the maternal immunity into the baby piglets through colostrum, and at the same time be able to vaccinate them. And one of the advantages of oral nasal vaccines is we have vaccines that do have the ability to overcome maternal antibodies if you can deliver them through the intranasal route. So some potential positive outlook on this particular coronavirus in pigs. So one of the things then that, as I mentioned in the beginning of my talk, where did this virus come from? How did it get introduced into the United States in 2013 so suddenly and so rapidly move around the country? And several veterinarians, private practitioners, and researchers were very much interested in this introduction, and they wanted to understand what was actually going on, because it was very curious how it seemed to be appearing across the United States very rapidly. And so the question began, what about this question of transport and feed, and would that have possibly been a route? Well, some of the first steps that were taken, actually, Dr. Scott D. Began pursuing this question. One of the things that triggered his effort was through farms that he was involved with in January of 2014 became infected within 1 week period of each other, and it was considered that those farms where those sows were located had very high biosecurity practices. The air was filtered for the barns that the sows were housed in, and good entrance-exit protocols were in place for the workers. And oddly enough, the clinical signs of PEDB on those sow farms where this was a clinical problem began about 1 to 2 days after a feed delivery onto the farm, and the consumption by the animals in the farrowing facilities. And the filling of the feed bin a day or two earlier appeared to correlate to the onset of the clinical signs. So they set about to try to demonstrate the role of feed as a way that this was being introduced onto these farms. And it was challenging to demonstrate. Some of the first things they did was they tried to sample various points on the farm, and they looked at dust particles from the exhaust fans on the barns to the ground below them, various concrete surfaces at various access points into the barns and around, sampled farm personnel as potential fomites, vehicles that were coming in and out of the area, both the vehicle cab as well as the tires, the feed truck driveway entrance into a feed mill, the exterior of the truck, and then interior walls of the suspected feed bins that were used to feed the sows. And this work has now been published. It's cited here, but they were able to go on and establish a proof of concept that the porcine epidemic diarrhea virus transmission very much was happening through the consumption of contaminated feeds. And so the risk of the feed as a vehicle, getting it introduced onto the farm is very, very real. Now, what that does is it means that there's various mechanisms that producers have to pay attention to when it comes to protecting the health of their herd. And so both contaminated feed ingredients now became a big concern, as well as other sources of environmental contamination, which would include the personnel going in and helping care for these animals. So the next step, now that they had some evidence that demonstrated that indeed the feed ingredients were a problem, particularly where they were under typical wintertime ambient weather conditions, that was all reported by Scott D and his colleagues, then this idea of an international feed source came under more scrutiny. And this paper here is one that is all published, and it's one that will describe in much more detail what I'm going to summarize for you here. But the original PED virus that was detected in the US, since it was extremely closely related to Chinese variants, it very much raised this question about the feed sources. And survival of the PED virus was demonstrated in soybean meal for as long as 180 days, as well as evidence of virus survival in other feed ingredients such as lysine hydrochloride, choline chloride, dried distiller grains, and several other items that might be used in feeding of pigs. And just to support the idea that this is a very real risk, you have the situation that there's a lot of feed ingredients that are imported into the United States from China, and this is derived from the U.S. government's harmonized tariff schedule from 2012 to 2016. And you can see that there are very large amounts of various products that are imported into the United States. And soybean meal became one of considerable interest for the rest of this story. So Scott and his colleagues set about to try to connect the dots, so to speak. And on this left-hand picture, you'll see the typical conditions in China where the grains are dried. They spread them out onto the ground and the roads and let Mother Nature and the sun do the best job possible at removing excess moisture so that these grains do not mold. And then they would get them transported to their ports, then onto a cargo ship, come across the ocean, and then be distributed once they get into the United States to the large hog production areas. Now, what Scott and his colleagues were able to do was establish the timelines that these various steps were involved with, and based on a commercial website that gave them the representative routes and timetables for movement of these products from China to the United States, they were able to show that a typical transportation route would be about 37 days in duration. So then the next thing they did was they set up environmental chambers and again, relied on data that was in the weatherunderground.com website, and actually mimicked the weather conditions, both temperature and relative humidity, with environmental chambers dating from December 23rd of 2012 through January 28th of 2013. So a time frame that preceded the initial detection of PED virus in the United States. And they programmed these environmental chambers to do the best possible mimicking of what a product was going to be experiencing en route to the United States. So here's a table of some of their results, and they've actually got a couple of published papers on this. Initially, they were looking at PEDV. This table shows a much broader collection of animal viruses, several of which are of major concern to U.S. Livestock producers, because some of these viruses we do not have currently in the United States. But I've highlighted here the column, the fourth column over here, with porcine epidemic diarrhea virus with the yellow circle around it. And you'll see that soybean meal in the red here, what that indicates that if it was red with a positive sign, that the infectious virus was detected in these feedstuffs after the simulated 37-day transport across the Pacific Ocean from China all the way to Des Moines, Iowa. They did a similar model indicated here in the title of this slide, where they did a transatlantic transport from Europe to the same point in Des Moines, Iowa. And that transportation model was 30 days in length. But they would spike these feedstuff or ingredients at the beginning of this model that they did in the environmental chambers. And then at various points in times, they were sampling them at the various steps. And these were the ones that made it all the way to Des Moines, Iowa. And so soybean meal, conventional and organic soybean meal sources from China did a pretty darn good job of supporting the survival of porcine epidemic diarrhea virus. There were other ingredients here as well. So again, I just put up here their published paper on this topic for people to get a much more in-depth description of what they did. I think it's a very, very important body of research that has identified some very serious concerns that we have for keeping animals healthy. So to summarize this body of effort, porcine epidemic diarrhea virus and feed, they have some very distinct links. Clearly within our country, once the region became contaminated, was not very long before the virus was being moved around the country with feed trucks. But obviously, their investigations identified the risk of feed as a vehicle for its introduction, as well as its subsequent spread throughout the United States. The PED virus isolate was 99% similar at the nucleotide level to Chinese strains, and it survives for weeks in feed ingredients under the transboundary transoceanic transport model that Scott D and his colleagues worked out. Infection can result from the consumption of feed through their normal feeding processes, and the oral infectious dose in feed is extremely low for porcine epidemic diarrhea virus. So very clear evidence that feed is a source for on-farm introduction here. So what has the pork industry done about this? And the pork industry involves obviously the National Pork Producers Council and then the veterinarians that support that industry. And they've been working to change some of our policies here in the United States. And they passed some resolutions this past year that were passed unanimously by the National Pork Producers Council to try to strengthen our efforts to prevent foreign animal diseases from entering the United States. So they had 3 separate resolutions were adopted, and these have been communicated up to the leaders of the USDA and the FDA on restricting imports of soy-based animal feed products from countries that have a high risk of foreign animal diseases in their country or they're present, which pose a risk to transmitting those pathogens to our livestock producers here in the United States. The second resolution was urging USDA and other private and public research institutes to continue evaluating foreign animal disease virus viability in pig feed and feedstuffs, and to develop hold times for imported feed where the virus may die off before these feedstuffs ever reach a living animal to be fed. And then the third resolution is to work with the National Pork Board, the USDA, the Food and Drug Administration, and the Department of Homeland Security on coordinating, along with Canada and Mexico, the development of practices to protect the North American swine herd from these foreign animal diseases. So I want to conclude with this point. Biosecurity is very important for food security. When one virus, porcine epidemic diarrhea virus, got introduced into the United States in 2013, in less than a year, it killed off 10% of our swine herd. Those are things that hit the pocketbook of producers, but they obviously impact the food supply in our country. And when you have a naive herd, you can very quickly have some serious food supply issues. The food safety was not a concern, but just the volume of animals that were dying as a result of porcine epidemic diarrhea virus was a very clear example of why it's very important that we prevent the introduction of foreign animal diseases into the United States. So with that, if there are any questions, Phil, I'd be happy to attempt to answer them.
Fantastic talk, Mark. Thank you very much. I mean, one thing that strikes me listening to you is that you surely would not have been one of the people who was surprised by the evolution of coronaviruses to infect humans as well, because it looks like you've been through this several times in animals, even very recently.
Absolutely, Phil. I think it's obvious. You got a very large RNA virus, one of the largest genomes of all the RNA viruses. They replicate or reproduce with low fidelity, meaning that there's an error rate, and it's more of a, if you will, a cloud or a mixture of viruses that get shed when you have an outbreak in a population. And so, no, the evolution of the viruses are constant, and Mother Nature is always out there trying to outsmart all of us, and very successful in doing so.
Yes, for sure. So in the context of the PEDV, I was very struck by that epidemic curve where you showed a rather substantial reduction in the number of pigs that were in the herd in 2013, but it seems like things went back to normal in less than a year. And what do you attribute that to? It sounds like the vaccine wasn't ready yet. I guess herd immunity might not be exactly the right word, or as we use it in humans, but did everyone get infected, or were some pigs somehow already somehow immune or protected from infection, or how was the industry able to bounce back so quickly? And are they still losing pigs to PEDV?
So I'll take your last question. I'll take your last question first. Are we still losing pigs to PEDV? That the short answer is yes. Not at the same rate we once were, and it's indeed due to the fact that there is a widespread herd immunity to it, the virus. One of the advantages of pigs is typically producers, a given sow can farrow, or when she gives birth, it's called farrowing. It's not unusual for her to wean 12 pigs or so per litter. And so the one advantage of the pork industry is that they can very quickly recover, but they've already taken the financial hit. So the other aspect of the swine numbers there is that a very large percentage of U.S. pork is exported around the globe. And right around 30% of U.S. pork is actually produced for export. And so the growing demand for U.S. pork over that time frame has contributed to the increase in the numbers of hogs. So there's a couple of factors in play there. And clearly there were some vaccines that did come out. We got some fairly rapid, I don't remember off the top of my head the timeline, but there was a vaccine that became available fairly quickly and was begun to be used by producers. But we have some different processes in veterinary medicine that we typically evaluate the host animal first because we can, which gets us to a better understanding of safety and efficacy of a vaccine much more quickly than what we are able to do with human vaccines. And I think herd immunity might have actually been derived from veterinary medicine and livestock production, because we have a lot of animals that are in herds. So I'm not sure which came first there. It could be a chicken and egg question.
Thank you very much. So there have been a number of questions about the thermostability of this virus, the PEDV, because it seems as though it really survived impressive temperature excursions that one would hope that a coronavirus wouldn't survive. And in fact, some of the feed may even have been heat treated. And so along with that, there's the question of what can you say about its thermostability, as well as is it possible, or would it be feasible if one wanted to improve biosecurity related to food stuff for animals, feed for animals, to do something to treat the food before it's given to the animals, or is that too impractical?
Well, that's an excellent question, and the people that are looking at these very questions are trying to find things that are practical to do. I would point out I went back to this slide that shows the upper line is relative humidity, not temperature. So the temperature line is this blue line on the bottom, and during the transport, you can see that this was actually fairly low temperature ranges that they had, that these things were that the products were exposed to that have been spiked with the virus. It is an envelope virus, so it's the seasonal pattern that we saw, where it survives more in the winter, is consistent with the heat lability of the virus. So I don't think it's anything too different than if you will, an average coronavirus. Certainly seems consistent with those that we see in livestock that have cold weather peaks and incidents and causing disease, and tend to be much reduced during the warmer weather. So I do think that heating is something that's going to reduce the environmental load, if you will. The people that are looking at some of the processes they might use to control the survival of the virus in these feedstuffs, they're looking at various ingredients that they might add that would diminish the ability of the virus to survive a 37 or 30-day transoceanic transport to help mitigate that risk. So many different mitigants are being examined right now. I think they're identifying some things that are helping. I'm not sure they're at a point where they have something that completely mitigates, but there are various companies that are involved with these scientists, and I think eventually we'll get to a point where they have identified mitigants that can be used that will hopefully dramatically reduce the risk of these items being brought into the United States through importation of livestock feedstuff ingredients.
Fantastic, thank you. Can you comment a little bit about testing of imports, or perhaps even food that's already here in the U.S. or livestock? What's the role of that? It strikes me as that perhaps as you do testing, you find something, you already know that you're behind the problem, although maybe you've caught something. Unless you're testing everything, you might be missing some things too. And one of the questioners actually asked specifically whether it's true that testing caught African swine fever virus contaminated pork in Long Beach. But if you could sort of talk more generally about testing, I think that would be very helpful.
So as a general rule, the feedstuffs you're literally are looking for a needle in the haystack. When you got, metric tons of material being brought in, there's no way that you can test them adequately to prove that every ounce of those metric tons, if you will, I'm blending English and metric units here, but it's very difficult, virtually impossible to prove that negative. So that's why, the idea is to identify mitigants that will be highly efficacious. In terms of the testing that's done, the questioner asked around African swine fever virus, we basically stopped the introduction of any fresh pork products in the United States at our various ports of entry. And I'm not aware of any actual testing. They just intercept all products. You're not allowed to bring them in. They're illegal to bring in. So that's the approach that is taken there. On the data that I showed you, these were deliberately spiked samples where they knew how much they put in and were able, under very controlled conditions, examine it. But these would not be practical approaches to try to go in and test all of the imported materials.
Thank you. What do you think we can learn for human vaccine development from the animal story? Of course, the goals and testing methods are different. Easier to do challenge studies in animals, and perhaps in the context of commercial pork production, the duration of immunity doesn't need to be as long. But where do you see the parallels and differences?
Well, I think that if you take a look at the pig, and there's a lot of use of the pig in human research, particularly in xenotransplantation efforts. The pig has a very similar physiology, a fairly similar immunology. I mean, there are differences, but they do represent an excellent model. So I think things that can be learned are along the lines of, okay, so we, along with the folks at Loyola University, have identified a way to very nicely attenuate the porcine epidemic diarrhea virus. The colleagues in Loyola have looked at some of the other viruses that are of interest, and the mutations made in the PED virus were orthologous to what they were looking at for some of the other important zoonotic coronaviruses. So I think that there's a lot of parallels that can be drawn, and I think there's some excellent research that's going on already in those areas. And I think the pig can just provide some additional evidence that these approaches may work. Now, one of the advantages, I'm a big proponent as an immunologist that if you have a disease that affects the mucosal surfaces of the body, that in veterinary medicine, the diseases we have there that we have a few very good vaccines for, they're delivered via the mucosal route rather than an injectable route. And we try where possible to develop vaccines that can be provided in that method to stimulate strong mucosal immunity so that you can not only have systemic immunity in the form of IgG, but you may have some very good mucosal IgA as well as mucosal cellular immunity that greatly reduces the capacity for a virus to successfully cause disease in an animal. So if we can take similar approaches in humans, if they can draw from anything that we get as far as insights there in veterinary medicine, I think that's all good.
So some of our listeners noticed the table that showed all of the different things that were tested for the ability to permit these viruses to survive, including pet food and other substances as well. And so I guess in this situation, it seemed likely that it was the soybean meal that was problematic. But for instance, you have the moist cat and dog food on that slide, even dry dog food. So what can you say? I guess should it sounds like you can't control this just by eliminating one of these types of imports, because others could actually bring these viruses in. So what can you say about that, as well as then also any identified risks and what should be done about potential risks, for instance, for domestic animals or other animals as well?
Well, that's a very good question, and I'm not sure that we're in a position to give a very good answer just yet. But clearly, identifying these as potential sources, all the viruses up here are mainly ones that affect livestock species, but they do have the capacity to survive in many instances here. So, I do think that just being aware of this and to be cognizant of where you choose to purchase your feed ingredients, particularly in livestock. Obviously, the pork industry has been fully made aware of these data. And as you saw with the resolutions that they have passed and have presented to several departments in the U.S. government. They're trying to make every effort that they can to minimize these as potential risks for them. As far as cat and dog foods, I'm not sure why they included all of these on here other than just as examples of products that were being imported. And, you know, and I'm not trying to point at any one country that we're importing from here, but these are just some examples that were driven by the fact that PED virus had such a high identity to the same virus in China. So I'm not really giving you a clear answer on that. But I think that certainly the pork industry is working to avoid importation of these products until such time mitigants can be identified that can be added in that are safe for animals to consume.
So can you say a little bit more about how you think the PEDV actually got into the food? So you think it's just because the drying of the food or the grains is done outside where animals might defecate on it, or are there other procedures that can be required to reduce the likelihood that that happens in the first place?
Well, obviously limiting wildlife access and animal access to where they dry them down would be a big step, but that is what is believed to be the method that it may have been introduced. There's other possible cross-contamination points as well, but you know, just differences in grain production systems. We don't do that in the United States or in Europe. So, you know, I think that it's a unique practice there, and perhaps they will be adapting what they do. I can't speak for their agricultural practices in that regard, but I think it quite clearly is believed by the veterinarians and the scientists that have done this work that was originally contaminated because there's significant feral hog populations in that area. And so that's one of the big reasons they were looking at, does African swine fever virus survive any of these feed ingredients being imported in the United States?
Thanks. What can you tell us about the pathogenicity of these viruses in their animal hosts? It was striking how the TGE, for instance, when it mutated to PRCV, actually went from being a virus that killed a lot of animals to one that was more likely to be subclinical or mild. And of course, as we're thinking about the SARS-CoV-2 and the situation in humans, it looks as though there's, well, we're dealing with viruses that can cause much more severe disease as these jumps are made. So any insight from the sort of the aggregate of the animal data that we can take on the severity of the disease and these viruses and how that seems to evolve?
Well, I think it's, you got to realize that viruses, and I'm sure you're aware of this, for a virus to survive, it's usually in its best interest to not kill its host. And if it's very good at killing its host, then its replication efficiency or reproduction efficiency in a population is dramatically reduced. So many of these viruses in veterinary medicine primarily have high mortality rates in younger animals, you know, baby piglets, for example. So if there's anything to be drawn on there, that's a little bit different than what we're seeing currently with the SARS-CoV-2. You know, with the bovine respiratory coronavirus, again, that's a very mild disease typically in cattle. It has some seasonal patterns to it. It can cause both respiratory and enteric disease in cattle. And research at our facility by some of our scientists, Dr. Julia Ritpath and Dr. Shali Falkenberg, they have been able to demonstrate that by itself, that virus is very difficult to get a disease model even established. But if you previously expose animals to another virus, one that's on this slide here, bovine viral diarrhea virus, which is actually primarily a respiratory virus in spite of its name, but that BVD virus is fairly immune suppressive for a period of time in cattle, and it can make the bovine respiratory coronavirus disease more severe if you infect the cattle first with a BVD virus for several days. So that's an example of a comorbidity, if you will, but an infectious one. So there's some things that we can learn about the coronaviruses in livestock, but I think there's some very unique aspects about SARS-CoV-2 that still a lot of work remains to be done.
I'm not surprised to have gotten a question about the tiger in the Bronx Zoo. And the question is sort of related to the issue of whether this might have not just been a spread to the tiger, might have involved a jump in species barrier. And then that, of course, calls to mind the overall concern about a virus that is spreading so rapidly as the SARS-CoV-2, whether that could, in fact, jump species barriers even to livestock. So can you sort of comment on this, the One Health aspects of this? How does this all fit together, and how do we make sure that what's happening in animals doesn't affect humans, and what's happening in humans doesn't affect animals?
Well, that's a very good question, and one of the best recommendations, if we want to protect our animals, if you're sick, please minimize your direct contact with your pets or your livestock. I'll use the example of influenza virus. There's actually a significant number of introductions of human influenza virus into pigs every year around the globe. And there's actually probably more examples of that being a reverse zoonosis than the other way around. So animals can be victimized by pathogens that we have as much as the other way around, if not more. And I think that if you are indeed exhibiting symptoms that are consistent with COVID-19, I think you should restrict your access to your pets. Wash your hands if you have to handle and care for your pet. Wash your hands before you do so, and again afterwards, just to make sure that you're minimizing their exposure. And if someone else can care for them in the household, that would be a good idea. There has been one study conducted in Harbin, China, that was released about a week ago in a preprint publication, where they had experimentally infected cats, domestic cats, a couple of different projects they did there. And they were able to demonstrate that the SARS-CoV-2 virus will infect and replicate in cats. And in their conditions that they studied this scenario, the cats did transmit the virus to other cats that were put in separate cages, but would be amenable to aerosol transmission between cats. So that work is waiting to have its peer review process completed. But that was some of the very first work that demonstrated that domestic cats could indeed be infected with this virus, done in Harbin, China. They also examined whether or not the virus could infect, replicate, and transmit in pigs, and they found no evidence that it could do that. So that was very good news if you're a pig. Our colleagues in Germany at the Friedrich Loeffler Institute, led by Martin Beer and several of his colleagues, they too have looked at experimental infections of SARS-CoV-2 in fruit bats, ferrets, pigs, and chickens. And very similar to what they found in China, China also in Harbin had examined whether or not ferrets would be a potential animal model for studying this virus. And the Germans were able to confirm that indeed ferrets, just like they found in Harbin, China, did support the replication of the virus. So a good potential animal model there, whereas the pigs and chickens did not. And that was similar to what they found in China, that chickens and ducks did not support the replication of this virus. So right now, I think that the best advice is, and China also looked at dogs in Harbin in their study and did not find that the virus was very successful in dogs either. So if you're sick, don't be handling your pets, don't be around livestock if at all possible. That's always a good practice to protect our animals when you have a zoonotic, potentially being infected with a zoonotic pathogen. And right now, it does not appear that SARS-CoV-2, we don't have any evidence to suggest that our livestock species at this point in time have any evidence of being infected by that virus.
Thanks. That's a great answer. In developing vaccines for humans, one of the concerns that has come up is this question of enhanced disease, this idea that previous immunity or previous infections might make a disease worse, and the risk then that vaccinating a human might make the disease worse if the type of immune response induced isn't strong enough. And so it was interesting to hear you mention that, I think you said BVDV makes BRV, prior BVDV can make BRV worse. And I know there's also the example of feline infectious peritonitis virus, where I think in the animal world, the discovery was made that an early vaccine for that actually made the disease worse in cats that got infected. So can you comment on any extrapolation between the animal situation and the human situation in thinking about this enhanced disease question in vaccine development?
So right now, I mean, good question, but one of the things that happened with the feline coronavirus was it went from being a virus that had a tropism for your enteric tract and respiratory tract, and then it changed due to mutations in the virus to infect macrophages and began to attack the immune system, and that's where the feline infectious peritonitis came along. So that one's a little bit different. Again, it was an example of a tissue tropism change. In terms of enhanced disease, we do have examples of vaccine-associated enhanced disease with some of our livestock diseases, but the BVD and bovine respiratory coronavirus example I gave, those were two fully virulent viruses, and the first one, being BVD, basically set the animal up to have a compromised immune system. So that, like I said, that was an example of a comorbidity that if you have impaired both innate and adaptive immune function that BVD can cause, that a more mild virus under normal circumstances can cause a more severe disease. So that's all quite different from, where you might get an enhancement of a disease as a result of some aspect of the immune response contributing to a more severe disease with a subsequent exposure. So I don't think we have any evidence at this point in time that would support that being a concern. But that's obviously why we do all of our safety trials and the efficacy approval process for human vaccines. So it's very important that those studies be done.
Thank you, Mark. So we're down to the last 2 official minutes here, and we can go on a little bit longer with a few extra questions if that's okay with you. But what I do want to do is, before we officially end at noon, is to make sure that the listeners are aware of tomorrow's webinar, which will take place at 11 o'clock. And our speaker there is Dr. Barney Graham, who's the Deputy Director of NIH's Vaccine Research Center. And he'll be talking about what we're learning from all of this, as well as, I think, some important aspects of vaccine development that he's learned. So I think that will also be an exciting talk, and I encourage those who are listening now to also tune in for that at 11 o'clock tomorrow. Another question for you, Mark, though. Is there anything we have learned from veterinary virology that may be helpful in developing antiviral drugs that might be applicable to these viruses as a class or to SARS-CoV-2 in particular?
So if I understand your question, what drugs might we be using as antivirals?
Yeah, in fact, are there antivirals that are used in the veterinary world or they're tested in the veterinary world that might translate to something that could be considered in humans, I think is the real question.
Yeah, if there's anything being used, it would likely be derived from human medicine. The situation for veterinary medicine with antivirals is very, very limited. Really use antivirals to any great extent in livestock at all, mainly because the process of getting an FDA approval, the FDA has pretty much wanted to make sure that antivirals are being used in human medicine because, as you well know, viruses mutate faster than bacteria, and we don't want use being used in veterinary medicine that would contribute to the development of antiviral resistance for the human population.
Excellent points. And as far as the virology from all of these animal viruses, which probably the study of which probably isn't as well funded as that of human coronaviruses, but as far as you know, are there any insights or Achilles heels identified in these viruses that might be seen as possible targets for coronavirus antivirals?
Well, clearly the example like that Drs. Fauberg and Baker have been doing together, they've been able to identify some of the non-structural proteins that interfere with the host innate immune response. I think those are great examples of what can be learned and maybe applied on the human side. Obviously, studies need to be done, but again, if those vaccines prove out to be highly efficacious when given through the oral route, again, that anything without a needle is always good, and particularly when you're dealing with food-producing animals. And hopefully some of the work there will prove to be very fruitful for the livestock producers. And if that can be an example of how to do things on a basic virology perspective, I think that they'll very quickly adapt that. Clearly, the molecular virologists have developed incredibly powerful tools to go in and inactivate these viruses in ways that 40 years ago we might only dream about. And so I have a lot of confidence in the scientific community and their ability to respond very quickly today. And then it's just a matter of demonstrating the safety and efficacy of what they're able to develop.
Great. Thank you. So I'm going to close with one more, I guess, more detailed vaccine question where, and of course, you did answer this general question about extrapolating vaccine information from the animals to humans. But one of our questioners is asking more specifically about live and perhaps inactivated vaccines and how they might be used separately or together in prime boost regimens, and whether there are learnings there about what yields the best immune response for any of these animal diseases that might be applicable to humans.
Well, that's a great question, and it seems that we need to do a lot more research on this area. A lot of the coronavirus vaccines that are used in veterinary medicine are modified lives that are attenuated, obviously. So, I think, and in veterinary medicine, some modified live virus vaccines are actually even approved for use in pregnant animals. So that's always a safety question, that you want to make sure your vaccines are safe for use in pregnant animals. So if there's similar approaches in human medicine, I think obviously you're going to have to do those studies in the target species eventually. So I don't have a good answer, but I have my personal view is that it's always good if you get both cellular and humoral immunity going, and your best ways of getting that done is typically either through live exposure with the virus or exposure to an attenuated or modified live virus. So that would be my personal preference of how to begin a vaccination program. And if you can do the priming that way and boost with inactivated vaccines, that would all be good. Clearly, we had only a few viruses that have been eradicated from Earth. I think the number is 2. And both of those involve the use of a form of a modified live vaccine, that being smallpox and rinderpest virus in livestock in ruminants. So I do think that the live attenuated vaccines will typically give us the best possible immunity that we can get if indeed we can demonstrate their safety.
Well, thanks again, Mark. I think we're now 5 minutes past the hour, and so probably a good time to close. But I'd really like to thank you for such an engaging presentation. It really is fascinating for someone like me who thinks more about the human side of things to see how you and the animal side end up struggling with exactly the same things, and in some cases get there first. And so I think that this presentation and this discussion provides us all with a lot of food for thought, with no pun intended. And I really appreciate you both putting together such an interesting presentation, as well as taking such a wide range of questions. So thank you very, very much. And we greatly appreciate your sharing your knowledge with us today.
Oh, my pleasure entirely. And just to emphasize my last slide on the presentation is that it's very important that our biosecurity practices help ensure a safe global food supply.
Well, fantastic. Wing, would you like to come in with any final remarks for the audience?
No, just to thank everyone, and of course the two of you as well. It's no easy job to be taking so many of these questions and answering them, but also to our audience as well. They've given us some stimulating questions today, so I do really appreciate that. And as Phil, you already mentioned, we do have Dr. Barney Graham tomorrow, same time tomorrow. So hopefully a lot of you can join us again for our third COVID webinar of the series.
Well, thank you very much for inviting us, Wing. And I think we all have work and other lives to go to. And so I appreciate especially the audience tuning in and staying on so long as well. So thanks, and I look forward to participating tomorrow again as well.
Thank
bye.
you, everyone. Thank you.
Bye all.